Hi, my name is Mark, and I'm going to demonstrate how to use the IC, the ion chromatograph. So what we have here are just some modules that control the eluent flow rate. We've got some pumps, the detector, a lot of control panels. We have the sample injection port right here from which you can introduce your sample. Behind it's not being seen, but there is the analytical column where separation of your analytes are going to take place. And we have the desktop computer, which has the instrument's operating software to integrate any detector output in the form of a chromatogram. When you come into the lab, you will the instrument will already be set up for you to use. Not much interfacing with the instrument is required except for the actual injection itself. So when you come in, you should be able to see this window on the display right now where it's asking you to click OK. When you see that, that means the instrument's ready to perform an injection. So I'll just move on to getting an injection performed. You're going to want to acquire a syringe just like this. So just make sure that there is a needle and a filter. The needle so it can enter the sample injection port, and the filters to prevent any particulates from entering the chromatograph. You want to make sure that the syringe has been properly cleaned before you do an injection of any contaminants from any previous experiments. So you'll just simply rinse it first with water and then with whatever you're going to be injecting into the instrument. So I'll first clean it out with some deionized water. You want to make sure when you clean it, you remove the needle and the filter when taking in sample. Then you can reapply the filter and needle, and then just dispense of the water into a waste beaker. And you'll want to repeat this process 1 to 3 times just to ensure that it is cleaned. And then with your sample, just want to repeat the same process. Again, remove the needle. And once the needle is sufficiently cleaned, then you can just take in sample into the syringe one last time. And then just reapply the filter and the needle. And now you're ready to perform an injection. To perform an injection, you can just simply place the needle or the syringe into the sample injection port, making sure that the dial is set in the load position. The sample loop behind the injector can hold a volume of approximately 50 to 100 microliters. So you want to ensure that that sample loop is filled with samples. So best to pump or to load approximately 1 ml of sample will ensure that that sample loop is filled. So I'm just pumping in about 1 ml, which is about 1 centimeter's length in the syringe. And to start the injection, I'll just simultaneously turn the dial from load to inject and click OK. And if you look on the screen, you can see the start of your baseline just forming. So the process of turning from load to inject starts the injection process and the separation process of the chromatograph. Also sends a signal to the software to start integrating, and you can now see the baseline forming. If you look on the screen, you can also see some additional information. It's telling you what sample is being run. It's telling you how long the run is going to be. It's approximately 11 minutes, and how much time has elapsed in the run so far. If you want to see what order your experiment is going to be performed in, when you come in, the experiment will already be programmed in a certain order, so you can see what sample you need to run next. So it's asking to run your 3 unknowns first, followed by your standard next twice, and then you're going to do another standard twice, and then just an unknown tap water solution. It'll tell you which one is running. The first one is highlighted, it's running. That tells you that that's what's being injected right now, and all the other ones are just waiting in line. If you go back to the screen, you can still see the baseline being formed. After about 30 seconds or so, you can turn the dial back into the load position, and you can remove the syringe and get it set up for the next injection. You can just see a dip in the baseline right now. That just shows that the deionized water band has passed through the detector. The deionized water has a low conductivity, so you're seeing a negative signal. And eventually you'll see the formation of peaks as your analyte is passing the conductivity detector. If you want to load up a chromatogram, you can just simply go to the browser window. You can click on one of the files. You can see it's already loaded up here. You can just simply go to the icon labeled right here, and it converts it into an Excel type file. You can then just save this by going to the Save As function, and then just pick a file name, and it saves it as an Excel file, and you can then export it, and you can print it out to another computer if you wish. So each run you're looking at about 11 minutes. So the best way to perform this experiment is to always have the instrument running, never leave it idle. Plan your experiments so that you're never leaving this thing idle. So while it's running, you're preparing other samples, just so you can finish this experiment more efficiently. And that's pretty much it for the IC.